Welcome to this lecture on mass spectrometry in computational biology. Today, we are going to explore one of the most powerful analytical techniques used in modern science. We will learn how researchers identify and measure the exact proteins functioning inside our cells. By the end of this video, you will understand both the physical principles and the mathematical foundations that make this incredible technology possible. Let us start with a simple analogy to understand what mass spectrometry actually does. Imagine you are handed a complex, fully built Lego castle, and your job is to figure out exactly which individual blocks were used to build it. You cannot just look at it from the outside, because many blocks are hidden deep inside. Instead, you decide to smash the castle into smaller pieces, and then carefully weigh each piece on a highly precise scale. By knowing the exact weight, you can consult a master catalog and figure out the composition. This is exactly how mass spectrometry works in biology, but we use electric fields to weigh protein fragments. Now that we have an intuitive grasp, let us look at the formal definition. Mass spectrometry is a powerful analytical technique used to measure the mass-to-charge ratio of ions. In computational biology, the process begins by taking a protein and using enzymes to chop it up into smaller fragments called peptides. These peptides are then vaporized and blasted with energy to give them an electrical charge, which is known as ionization. Once charged, these ions are accelerated through a vacuum. Their flight paths are altered based on their mass and charge. Finally, a detector records when and where each ion hits, creating a spectrum that algorithms match against a database. You might wonder why we need this complex machine when we can just sequence a person's DNA. The truth is, having a gene in your DNA does not mean the corresponding protein is actually being produced in your body at any given moment. DNA is just a static blueprint. Mass spectrometry is crucial because it tells us exactly which proteins are actively present, in what quantities, and if they have been modified by the cell. In the real world, this technique is used every day to discover new disease biomarkers, such as finding specific proteins in a blood test that indicate early-stage cancer. Understanding the biological importance leads us directly to the core mathematical measurement of this technique. The mass spectrometer does not measure pure mass, it measures the mass-to-charge ratio. Conceptually, this is because the electric field pulls on the molecule based on its charge, altering how its mass behaves in flight. In this simple formula, m represents the mass of the peptide ion, typically measured in daltons. The variable z represents the integer number of charges on the ion, such as +1 or +2. When the mass increases, the ratio increases. But if the charge increases, the ratio decreases, meaning the machine feels the molecule as being lighter. The final unit is the Thomson. Before we move on to more complex physics, let us see this ratio with a concrete example. Imagine we have isolated a specific peptide fragment from a viral protein. We're given that the exact mass of this peptide is 1,500 daltons. During ionization, it gets stripped of 2 electrons, giving it a net positive charge of +2. We need to calculate the mass-to-charge ratio the detector will record. We simply divide the mass, 1,500, by the integer charge 2. This gives us a final answer of 750 Thomsons. Physically, because of the double charge, the electric field pulls twice as hard, making the peptide behave as if it were half its actual weight. Now that we have calculated a basic ratio, let us look at the actual physics of the machine in action. When an ion is accelerated by an electric field, the electrical potential energy given to it is completely converted into the kinetic energy of its motion. If we increase the voltage applied by the machine, the velocity of the ion will increase, meaning it will fly faster. Conversely, if the mass of the peptide is larger, it will be heavier and therefore travel at a slower velocity for the same amount of applied energy. In a highly popular type of mass spectrometer called a time-of-flight analyzer, we do not measure this velocity directly. Instead, we measure the exact time it takes for the ion to travel a fixed distance. This concept of measuring travel time brings us to the beautiful time-of-flight equation. Conceptually, this formula is derived from equating electrical energy to kinetic energy, allowing us to translate biological properties into a measurable unit of time. Let us break down the symbols. The variable t is the time it takes to reach the detector in seconds. The variable d is the fixed length of the flight tube in meters. Inside the square root, m is the mass, z is the integer charge, E is the elementary charge constant in coulombs, and U is the accelerating voltage in volts. Notice that if the mass m increases, the time t increases, meaning heavier ions arrive later. If the charge z increases, the time becomes shorter. Now that we have our formula, let us apply it to a real-world example. We are given a peptide with a mass of 3.2 times 10 to the negative 24 kilograms, a charge of +1, an accelerating voltage of 4,000 volts, and a flight tube length of 1.5 meters. We need to calculate exactly how long it will take to hit the detector. First, we carefully substitute all our known values into the equation. Next, we simplify the denominator, which represents the total electrical energy applied. Then, we divide the incredibly small mass by this energy value to isolate the mass-to-charge effect. Finally, taking the square root and multiplying by the distance gives us 75 microseconds. Physically, this means the computer measures microscopic fractions of a second to determine the exact mass. Having successfully used the time-of-flight formula, let us understand exactly where it comes from. We start by establishing the fundamental physics inside the machine. The electrical potential energy given to the ion, which is Z times E times U, must perfectly equal the kinetic energy it gains, which is 1/2 mv squared. Since we cannot easily measure the velocity v directly, we express velocity as the fixed distance d divided by the travel time t. By substituting this into our energy equation, we link the physical energy of the system directly to the time variable we want to measure. Expanding the squared term sets us up perfectly to isolate the time variable in the next step. Continuing from our previous setup, we now want to isolate the time variable, t. We algebraically rearrange the equation by multiplying both sides by t squared and dividing by the electrical energy term, ZEU. This gives us an equation for t squared. Notice how we can factor out the distance squared, d squared. Finally, by taking the square root of both sides, the distance d comes out of the root cleanly, leaving us with our final time-of-flight equation. The intuition here is beautiful. The time it takes to fly through the vacuum is directly proportional to the flight distance and scaled by the square root of the mass-to-charge ratio. This exact math is what the software uses backward to identify proteins. Now that we have proven the mathematics, let us look at a visual schematic of the entire process. On the far left, you can see the biological sample entering the ion source, where a laser vaporizes and charges the peptides. Notice the voltage plates in the acceleration region. This is where our electrical energy equation takes effect. As the ions enter the long vacuum flight tube, you can clearly see the physics we just calculated. The smaller, lighter ions are flying much faster and are ahead of the larger, heavier ions. Finally, on the right, they strike the detector. The computer at the bottom translates those exact arrival times into the mass spectrum graph you see on the screen, showing distinct peaks for each peptide. To wrap up our lecture, let us review the most important ideas we have covered. First, mass spectrometry is an indispensable tool that identifies active proteins by measuring their exact mass-to-charge ratio. We learned that this is achieved by ionizing peptide fragments and accelerating them through a vacuum. Second, the time of flight equation is the mathematical heart of this process, perfectly linking the physical mass of a molecule to the microscopic fraction of a second it takes to reach the detector. Finally, by matching these precise measurements against biological databases, we bridge the gap between the static blueprint of DNA and the dynamic, living reality of the proteins that actually run our cells. You now understand the core of computational mass spectrometry.